Chapter 9 covers what genes are. So in this chapter, we're going to be looking at our genetic material, which is DNA, and also looking at some applications of DNA replication and how it can relate to something called CRISPR, which is used to edit genomes that can be, for example, used to grow transplant organs from pigs for humans. So in order to understand what genes are, we have to take a look at our genetic material, which is DNA. DNA stands for deoxyribonucleic acid, and this contains the genetic code for all life. It was one of our 4 major biological molecules that we looked at earlier in the semester. This DNA molecule, it's a huge, meaning very long, double-stranded molecule made up of building blocks called nucleotides. Each nucleotide is composed of 3 parts. In DNA, it includes a sugar, specifically a deoxyribose sugar. To that sugar, there is a phosphate group attached. So we have our phosphate group and deoxyribose sugar. The third component is called a nitrogen base. And there are 4 possible bases that can be attached into this third spot. These 4 possible bases are adenine, cytosine, guanine, or thymine. And you can abbreviate them as A, C, G, or T. So these nucleotides are put together, bonded together into strands. And in order to get a DNA molecule, we have to have 2 parallel strands of DNA. These 2 parallel strands are connected by hydrogen bonds across from each other, and those hydrogen bonds link together those 4 different bases, so those nucleotide base pairs. And this is not just done randomly, this is done using what's called complementary base pairing, or Chargaff's rules, because they were discovered by a scientist named Chargaff. So these complementary base pairing rules state that adenine pairs across from thymine, or A equals T. We can see that here. In addition, cytosine pairs across from guanine. So C's pair across from G's. So using these base pairing rules, that is going to create this double-stranded DNA molecule that has this complementary base pairing. That double-stranded molecule will then twist due to how the chemicals are bonding with each other. And from that, we end up with a DNA ladder that's twisted into a spiral called the DNA double helix. And that is shown right here. So within our DNA double helix, all living organisms contain DNA that gives them that genetic instructions. The sequence of those bases, the adenine, thymine, guanine, and cytosine, it differs in every single DNA strand, and this can differ among different species. So for example, we have different sequence of DNA in humans versus domestic dogs versus different plants. In addition, it can vary within a subtype within a species. So for example, if you think about domestic dogs, we have different breeds of domestic dogs. And each of those different breeds, from Dalmatian to Great Dane, they have differences in that DNA that codes for those different traits or different characteristics that those different breeds show. And even individuals in the same species can differ. So for example, one Dalmatian dog can differ from a second Dalmatian dog. So they can have different weights, different heights, different number of spots, for example. So there's a lot of variation within these DNA sequences. So one thing that has to happen with our DNA is it has to be passed down from one generation to the next. So that is done. Oh, sorry, so this is going back. So remember, differences in our genotype or our DNA nucleotide sequence, this results in differences in the phenotype or those physical properties that I just kind of mentioned a little bit ago. So because of this, those slight differences in that nucleotide sequence of those two different alleles, so kind of relating it back to the genetics, this is going to cause slight differences in the protein product that's produced, thus the different phenotypes or the different physical traits and characteristics that are going to be created. So for example, if we have the genetic sequence down here of A, GGC, TTAGA, this gene or this sequence of nucleotides within the DNA, that's going to code for a baby blue pigment in some feathers. If there's a slight difference in that nucleotide sequence, so for example, we have A, G, G, C, T, and then we have A. So up in the top we had a T, now we have an A, G, A. This is going to create a slightly different blue pigment, so we'd have turquoise blue pigment in feathers. So these very tiny little differences in those nucleotide sequence of the A's, the T's, the G's, and the C's, how many there are, how they're ordered, that will cause different protein products, which will cause different phenotypes or those traits or characteristics. So now that we understand that these genes or these nucleotide sequences code for different phenotypes, we can use a technology called CRISPR. And this CRISPR mechanism, it's a natural defense mechanism in bacteria, but scientists have adapted it for laboratory use to edit the DNA or the genes of an organism so that they can produce a specific type of gene, which will produce a specific type of product. So here, for example, this is some genome editing using this CRISPR tool. So this CRISPR tool, it is an RNA molecule, and they're shown right here in these blue circles with the red RNA sequence. So in this case, this CRISPR mechanism is going to find a specific gene highlighted in the green on our DNA double-stranded molecule. That CRISPR is going to enter in, it's going to physically cut out parts of the gene. That gene can then be removed, and now, for example, this gene is no longer in those cells. So this is one way that we can edit genomes. We can cut out specific genes that code for specific products. In this case, they're cutting out a gene in the pig, or they're knocking out a gene and it remains non-functional so that organs grown in pigs will not be rejected by human recipients. So all this DNA that we're looking at that codes for this genetic material, it is condensed down into chromosomes. So what happens first is our DNA double-stranded helix up at the top, right here. This DNA, we have to condense it down to fit it into cells. So what happens first is this DNA, it's wrapped around proteins called histone proteins. And that combination of our double-stranded DNA and these histone proteins, that creates what's called a nucleosome. These nucleosomes zigzag back and forth to create what are called chromatin fibers. Those chromatin fibers compact down even more to make individual chromosomes. These chromosomes need to go through replication in order to be passed down from parent to offspring. So DNA replication, this occurs when we have duplication of a DNA molecule. And this occurs right before a cell enters into mitosis, so right before cell division. DNA replication occurs very rapidly when cells become cancerous. And it also occurs when viruses hijack a cell, and viruses can copy their own DNA. So DNA replication is very important for the continuity of life and to pass down that genetic information from one generation to the next. So what happens during this DNA replication? We have three steps that occur. The first thing that happens is that proteins bind to an origin of replication. This is where the DNA replication is going to start. These proteins unwind the two strands of the DNA in that double helix, so they break down those hydrogen bonds connecting those two strands together. So remember, we have two strands of DNA, and they're bonded together using those complementary base pairing rules. So if you remember, A pairs across from T, and C's pair across from G's right here. So what happens is those bonds between those two strands is broken by these proteins at that origin of replication. Once we have those two strands broken apart from each other, a special molecule called DNA polymerase, it's an enzyme, it uses each strand as a template to build another new strand. And in order to start that, we have to have a primer that's put on first. So it's kind of like if you paint a room or paint a house, you have to put on a primer before you actually put on the paint. So we have to put primers on that indicate to the DNA polymerase where it should attach on, and then that's when those complementary strands will be created. And this is done in a semi-conservative manner. So this is called semi-conservative replication, which means that there are two new double DNA helices. Once each of those two new DNA double helices molecules has an old strand and the other side is a newly synthesized strand. So we'll take a look and I'll show you what this looks like. Right. So up here we have our DNA double helix up in blue right here. So we have our origin of replication that occurs. We have a protein that comes in and separates those two complementary blue strands from each other. So you have an opening right here. DNA polymerase then comes in on each of those two blue strands and it adds in a complementary pink strand. So those blue strands are the old parental strand and the pink is the new strand. That new strand is synthesized using those complementary base pairing rules. So adenines go across from thymines, or A's and T's match up. Cytosines go across from guanines, so the C's and the G's also match up. Then we have that semi-conservative part. So if you look at each of these two strands that were just made, one part of it is blue, coming from the parental or original strand. The other side is pink, which is the new. So again, that's that semi-conservative. It's half old, half new. These two DNA strands that were just created are also genetically identical to each other. So this DNA replication, again, this is used right before mitosis. It's used before meiosis. So basically, anytime the cell is going to go through cell division, that DNA has to be replicated first so it can be passed on to those new cells. So scientists have figured out how this process works, and we can do this in the lab. And this is called PCR, or polymerase chain reaction. This is a really useful lab technique where we can collect DNA and force it to go through DNA replication. So this can produce millions of copies of that targeted DNA sequence in just a few hours. And this is used in crime scenes to help identify suspects and victims. This is used to do food safety testing. So for example, I worked in a lab that used PCR to test beef coming from the slaughterhouse for the presence of E. coli before it could be taken and ground up into ground beef. So this is a very, very useful technique. It's used in a lot of biology testing laboratories. And another neat thing about this is it can be started with an extremely small amount of DNA, and it can create a larger amount of DNA that we can actually identify or use to do a lot of different things. So what happens in the polymerase chain reaction, and again, this is just an artificial way for us to do DNA replication in a lab. So what they do is they collect DNA, they place that DNA into a machine called a thermocycler. And it's called a thermocycler because what happens is we take that DNA, we heat it up, that forces our original two strands to separate from each other. We then add in DNA polymerase, which creates a complementary side. So now we go from one DNA strand to two identical DNA strands. And that happens in what's called a cycle. So this thermal cycler, it heats up, cools down the DNA multiple times, so it goes through multiple cycles. And every cycle, we double the amount of DNA that's in that tube. So if you start off with one DNA strand, the first cycle, you'll have 2. After the second cycle, you'll have 4. After the third cycle, you'll have 8. And it will grow exponentially or double with every cycle that occurs. So you can see that with a very small amount of DNA, we can end up with millions of DNA sequences after like 40 cycles, or if it keeps going. So this DNA replication, one part about it is that we have to be able to proofread during that DNA replication process. So we have DNA polymerases that can proofread, or it's a DNA repair system. It can correct almost all the mistakes during replication. There are other proofreading molecules that are constantly reading over the DNA, correcting any mistakes, because we really want to make sure that that DNA has integrity, that there aren't any major mutations or errors that are going to be passed on from one generation to the next. So it can correct up to 99% of the errors. So what really happens is there's only one mistake for every billion bases within our DNA. So this proofreading is very, very good. However, that one mistake in every billion is still just enough where we can end up with mutations in the DNA. So mutations can occur from during this replication process. Mutations can also occur if the DNA is exposed to any mutagen like UV light or other chemicals. But we're going to be looking at it from doing a mismatch during DNA replication. So mutation, this is just a change in the sequence of nucleotides within an organism's DNA. Specifically, again, we're looking at what's called a mismatch mutation. So this is when we have an error during DNA replication that escapes that repairing process that I just talked about. So a lot of times this will lead to what's called a point mutation when only a single base is altered. And there's a couple of different types of point mutations, 3 specifically. So 3 different types of point mutations that can alter a DNA sequence. There's substitution. So this is when one nucleotide is exchanged for another in a sequence. So an example, if we have A, T, G, C, and we end up with A, G, G, C. Sorry, that's supposed to be a C right there. This is an example of a substitution. So instead of a T, there is a G put in place. So one nucleotide is exchanged for another. Another is we could have a nucleotide that's added. So if we have our original strand right here, A, T, G, C, if a nucleotide's added, we could end up with A, T, G, C. So this second A right here was inserted, it's an extra. Another thing that can happen is a deletion. This is when a nucleotide is removed. So for example, we could have A, G, C. So that T that was right here before, it was deleted out, so it's no longer there. So these tiny little mutations, I wouldn't think that they would make a big impact on organisms because you're just either switching one, inserting an extra, deleting one little nucleotide. But there are some big consequences that can occur from this. One example of this is sickle cell anemia, which happens with just one little substitution mutation. So in this example, normal hemoglobin, so hemoglobin is used to transport oxygen in our red blood cells. So we have a gene that codes for this protein hemoglobin. If you produce normal hemoglobin, your red blood cells will have a nice round shape to them. So that's over here on the left. If you have a little substitution that occurs within that hemoglobin gene, so just a tiny little switch from T and substituted with an A, highlighted in the yellow on the right, this tiny little substitution leads to what's called sickle cell hemoglobin, which leads to red blood cells that have the sickle or a C shape to them. So this tiny little change can influence the shape of your red blood cells. So again, some of these mutations can have very big impacts. And the big kind of impact that this has is normal red blood cells are very good at carrying oxygen and getting rid of carbon dioxide. These sickle cell red blood cells are very bad at carrying oxygen and getting rid of carbon dioxide. So this can affect your whole body, because if you're unable to get oxygen to all your cells will be unable to do cellular respiration and create ATP, that energy molecule. So you'll really struggle kind of not getting enough oxygen if you have the sickle cell hemoglobin gene in your DNA. So this chapter really focused on looking at genes, looking at that they're made out of DNA and how those genes or how that DNA can be replicated so it can be passed down from parent to offspring, and how tiny little mutations in the DNA can influence that phenotype. So this goes back to that genotype, or the genes that we have, or the alleles, those versions of genes that you have, can really influence your phenotype. Or any of those traits or characteristics that you show.